The DNA present inside can be copied into a small molecule called RNA molecule. It acts as a messenger, therefore we call that the name mRNA molecule, the messenger RNA. The mRNA molecule, since it's very small, it can pass through these nuclear pores. But remember, DNA can never leave out of the nucleus into the cytoplasm. Only mRNA molecules can. So when they leave, so the nuclear pores, usually those are connected with this rough endoplasmic reticulum. And RER, they have numerous ribosomes attached to their surfaces. The first thing, the transcription happens in the nucleus. That's what we discussed. The copying of DNA to mRNA is known as transcription and forms mRNA. The second phase happens in the ribosomes. We call this process translation. So the meaning of the translation is that the mRNA sequence is read by ribosomes and their proteins are made. Amino acids are joined together and those are linked by peptide linkages to make polypeptide molecules. And this process we call translation. This is where what happens in the ribosome.
Sir, can you tell about translation again?
Translation again.
Yeah.
Right. Yes. Now, so in the ribosomes, these mRNA strands are translated. So strands, those are read amino acid sequences are joined together. So with the help of small transfer RNA, we call tRNA molecules. Translation. So as a result, a polypeptide chain. You can simply call this the protein is made. So have an idea. So think how DNA are packed into chromatins and how those are arranged in chromosomes. And think of that how histone proteins and DNA molecules are wrapped around each other, and have the idea that mRNA molecules can be made by copying DNA, and the mRNA can leave the nucleus through nuclear pores, or the small gaps, holes present in the double membrane nuclear envelope. So they can go through this rough endoplasmic reticulum, and then it meets the mRNA molecule meets ribosomes, and the ribosomes is the place where the proteins begins. That means the second phase, the translation, takes place there. So as the mRNA molecule read by these ribosomes, these structures, new molecules called tRNA molecules, they come and join with these ribosome subunits. Now the tRNA molecules, they bring amino acids. Now we discuss mRNA has a code. That code, according to the code, so it is read as triplets, 3 letters. So the corresponding tRNA molecule with the complementary opposite codon, or the complementary opposite, the letter sequence, come and bind with this mRNA molecule in the ribosome, and each transfer RNA molecule bring an amino acid. Now, when two tRNA molecules are closer together, those amino acids are brought close together. Now, in this time, a reaction occurs where two amino acids are joined together by forming a peptide linkage. This process we call a condensation reaction, in where we get a dipeptide. So similarly, so many peptides bonds are made and many amino acid molecules are joined together, and we call this process now the production of polypeptide or the protein by translation, and that completes the process of protein synthesis. Now see here the transcription. So this is a word we discussed, that we have DNA. The DNA is a double strand. So at one point, can you see this double strand? It just opens up from the middle. So it loosens up from the middle and the bonds are broken in between those, the DNA nucleotides, between nucleotide bases, and an mRNA molecule is made. RNA molecule, we call it M because we call it a name, it's a messenger. You know what messengers do? They take information from one place to another place. The mRNA molecules, so then, so it has a sequence. Can you see there are some letters written here? So like that, it is a copy of that exact base sequence present in this strand of one of these strands. So that is copied and it goes out of the nucleus through the nuclear pore. Now, even though it's not shown here, so now it enters the rough endoplasmic reticulum and the ribosomes there. And the ribosome is not that large. This is just enlarged, right, to make it understandable. So then, so this is the structure, the ribosome. So this structure, the whole structure is ribosome. And there you can see the RNA molecule is red. So there are like letter structures, like you see, C, A, G, U. So these are we call nitrogenous bases. We learn them later. Those sequences are read. Now, according to, now 3 letters are read at once. Now, there are molecules called tRNA mentioned in the nucleus, sorry, in the cytoplasm. The tRNA, transfer RNA. So, each and every tRNA molecule has a joined amino acid in one end, and other end they have a 3-letter sequence that we discussed. So, there are complementary sequences to these 3 letters. So, they come and bind here. That happen according to pattern. When 2 of these tRNA molecules, they bind with this 3-letter codon on the mRNA, and then these amino acids are joined together. So now this makes a growing protein chain or the polypeptide chain, and eventually, after the end of the mRNA molecule being read, now it produces a protein, single protein. So this is the story of the protein synthesis. Once again, it's okay even if you don't understand. And we need only just the idea. So what's the importance of the nucleus, DNA, and the ribosomes, and rough endoplasmic reticulum? Because if I say you certain words now hereafter, it must be much clearer to you. Well, okay. So then, since we are talking about the cell organelles here, I'll give you the important ones. Right. So let's talk about this. Just write a nucleus. Nucleus. So right here often has a nucleus is often has a spherical or oval shape surrounded by a double membrane called the nuclear envelope, so which separates the nuclear content from the cytoplasm. The outer membrane is continuous with the rough endoplasmic reticulum that we call RER. Several tiny pores in the nuclear envelope allows substances pass between the nucleus and the cytoplasm. Remember that relatively small molecules can diffuse through the nuclear membrane. You know what diffusion means, right? And the larger molecules definitely they cannot transport, but certain smaller so molecules such as these RNAs and even certain proteins so can pass through that problem. Now we know that ribosomes are made in nucleolus, so those need to be exported from the nucleus. So now these ribosome subunits are moved out, right? So right here, mRNA molecules and also ribosomal subunits. They transfer from nucleus to the cytoplasm. And what are substances that come inside? Now, certain proteins should come inside. The amino acids should come inside. Nucleotides can come inside. Those things such as the nucleotides, amino acids, and even certain proteins. So they come from cytoplasm to nucleus to the nuclear pores. These things they move from nucleus to cytoplasm. Then still we are under the nucleus. Let's talk about nucleoplasm. It's a subtopic of the nucleus. Nucleoplasm. Now, when you take the nucleus, that we know it contains nucleolus, and also then we have this chromatin. So this we call the nucleoplasm. This is a nucleolus, and these are chromatin. So here we have both double membrane, right? The envelope, so that contain both, and there are pores here, the nuclear pores. So that's the structure of the nucleus. Right. So mention made up mostly of water. Mostly of water. A mixture of various molecules and dissolved ions. Right. It's a gelatinous and sticky liquid that supports chromosomes and nucleoli. Chromosomes and nucleolus that contain chromatin. Right under that, we'll talk about chromatin. Loosely coiled mass of threads. Loosely coiled mass of threads consisting DNA, histone proteins, and small amount of RNA. Right. These threads condense together to form chromosomes during cell division. So what chromosome means? A structure made of tightly coiled chromatin which is visible during cell division in the nucleus of eukaryotic cells. Now, once again, I'm going back to that picture I brought you before. Now, these are noodles we see inside the nucleus. Oil threads called chromatin. Usually, when you check under the light microscope, you see nothing. You just see the margin of the nucleus and the cell, so like that. And you don't see even like coil structures because this is like colorless. You can't see them. But remember, there's one stage of the cell. One stage of the cell. The stage of the cell is that where the cell is about to divide. We call it cell division. You must have learned about mitosis and meiosis, cellular divisions or the nuclear divisions. So when it's about to divide by this one of these processes, at one point the chromosomes, so this chromatin, they condense and thicken and become shortened. So then they become these structures and they organize into these structures. This is the condenser chromosome, and these are now known as chromosomes, right? And once again, later I will explain to you, the chromosome looks like this, and so these are called chromatids. I'll explain later. These chromatids are exactly similar copy of each other. And once they are separated from the middle, so they are now called chromosomes again. Okay, now we'll talk about the nucleolus. Next topic. The densely stained region of the nucleus, which manufactures ribosomes using the information in its own DNA. The mitochondria is the cell organelle which is responsible for the aerobic respiration of eukaryotic cells. So as you see, it's a double membrane organelle. The inner membrane is highly folded to form cristae. The fluid interior is called the matrix, and the matrix contains a small circular DNA known as mitochondrial DNA, and also it holds small 7ts ribosomes. The sizes of the mitochondria, the diameter varies between 0.5 to 10 micrometers, and they have specialized organelles to carry out aerobic respiration and hence to produce ATP molecules. Right. Let's write about this rough endoplasmic reticulum. Simply, we'll first discuss about the endoplasmic reticulum and then the rough endoplasmic reticulum. Flattened sheet-like structures called cisternae. They are continuous in the nuclear lobe, make up the endoplasmic reticulum. Right now, that's the rough endoplasmic reticulum. Let's say RER. So like that, covered with many tiny organelles called ribosomes. The site of protein synthesis. Site of protein synthesis. Proteins are transported to RER after the synthesis. Called SCR. So smooth endoplasmic reticulum, it lacks ribosomes, so it is similar to the endoplasmic reticulum, and it manufactures lipids and steroids, including reproductive hormones such as estrogen and testosterone, and molecules such as cholesterol. And smooth endoplasmic reticulum also is a major storage of calcium ions. Right, Golgi apparatus. The Golgi apparatus is a series of flattened membrane-bound sacs called cisternae. It constantly forms and breaks up Golgi vesicles at one end of the apparatus, collect processes and packages proteins and other molecules, particularly which comes from REA. Create secretory vesicles, and lysosomes. Then hope to see you next week then. Shania?
Okay, sir.
All right. Okay then. Have a great week. Enjoy. Good afternoon.